And you have me for 2 hours today. It's a long. So what I'm going to do, it's going to be an extensive lecture on CRISPR and human gene editing. And I would like to divide this lecture into 2 parts. So this is what I would like to do today. I would like to go over the history of genetic engineering in human beings. I will then review the science. What exactly is CRISPR and how does CRISPR work? And then last month, I had a chance to visit the 3 CRISPR companies in Cambridge, Massachusetts. So, and I'm building a relationship with them. And so what I would like to do is to talk about 2 of the 3 companies. The chief scientific officers of the companies were very generous, and they gave me their slides that they used when I spoke to them and when they spoke to me last month. So I'd like to introduce you to the industry. And then we will move to distinctions, several important distinctions. And then finally, we will end with a conversation about the ethics of CRISPR. And so we have 2 hours together. And so what I would like to propose is maybe after the industry, we will take a break for 5 minutes or so before jumping to the distinctions and ethics. I encourage you to raise your hands if you have any questions. I'm an American, I'm used to that. My students do that all the time. So if you have any questions while I am speaking, please raise your hands. I have also included a couple of slides that will encourage me to stop and to ask you what you think. And so I would like this to be much more of a conversation that you may be used to. So this morning I would like to begin with the history and with the history of CRISPR. And what I have done is I have gone online and I have looked for 2 timelines that illustrate the history of gene therapy in humans. So I'm going to begin, however, with definitions for gene therapy. Now, I would like to also let you know that I'm going to provide all of these slides as a PDF. At the end, Monday's slides and today's slides, I will provide to Father Joseph after my presentation as a PDF so that you can have those slides. I apologize for not providing them before the lecture, but I still was changing them on the train here, and because I'm still thinking about what I wanted to say. So I'll give you the slides after the presentation rather than before. So I'm going to begin with 2 definitions of gene therapy. The first is European, and the second is American. So here you have the definition from the European Medicines Agency for gene therapy. And you notice that there are 2 components of this definition. The very first is that the active substance must be a nucleic acid. It must be either DNA or RNA. And the reason for providing this nucleic acid to the patient is to change the sequence of the DNA or to alter the regulation of that genetic sequence. And I will review what a genetic sequence is. I will review what DNA is in a few minutes. And the purpose of providing this nucleic acid to a patient, as you can see, is for therapeutic, prophylactic, or diagnostic effects. So therapeutic is to cure, prophylactic is to prevent, and diagnostic is to figure out what is happening in the patient. So this is a European definition for gene therapy. We now move to the United States, and I'm here using the definition of the US Food and Drug Administration, the FDA. You can see it's very similar to the European model. First, you have to have genetic material, and the genetic material must integrate into some way into the host genome. It must be permanent in some way. This is the difference. Integrating into the genome makes it permanent. And you can administer it, you can give it to the patient as a nucleic acid, as a virus, or a genetically engineered microorganism. So very similar overlapping definitions in Europe and the United States for gene therapy. So I would like to begin now with a short discussion of the history of gene therapy. So the first is this timeline, which I took from a paper that was published in 2013. Yes, question. Can you? Yes. Here's the, let's see if we can use the mic. Okay, it's not on. Yep.
Honestly, it was a yes or no question.
Yes.
But is there any part of the U.S. definition that includes what it should be used for in the same way the other definition did?
Now, I think the idea is that the assumption here, because it's an FDA, so the FDA only deals with drugs. So the idea is that it's a drug of some sort, and drugs are primarily defined as prophylactic, therapeutic, and diagnostic. So I think that's assumed by the fact that it's the FDA that is doing it, not, say, for example, the EPA. So if it was the Environmental Protection Agency, then there might be other purposes associated with it. But because it's the FDA, the assumption here is that it's medicinal in some form. Hopefully that answers your question. Thank you. Okay, so we're moving now to this timeline of milestones for gene therapy. It was published in a paper from 2013, and it's important for me to highlight the date it was published because CRISPR was discovered in 2012. So the paper is published in 2013. It does not yet have a discussion of CRISPR. So what I would like to do here is to talk about the history of gene therapy immediately prior to CRISPR. So for a lot of people, you begin in 1928, because in 1928, Frederick Griffith begins to describe how something can change the behavior of cells. So here he talks about the transforming principle. The transforming principle is DNA, but he does not know it's DNA. And he can change viruses that are non-virulent to viruses that are virulent by simply transferring something from one virus to another. So in 1928, you have the discovery that something material from one cell to another can carry what we would call now information. The information in this case was the ability to produce disease. And the first 50 years of this timeline deals with advances in molecular biology, the discovery of DNA, the discovery of how DNA can be transmitted, and of course, significantly, the structure of DNA. So in 1953, Watson and Crick discover this double helical structure of DNA. And that is critical because it shows us that information can be copied and then given to the next generation of cells or the next generation of organisms. In 1968, Rogers and Pfafunderer describe how DNA can be moved from using a virus. It's very important. You have the discovery of the molecule that carries genetic information, and then you need to find a way to transfer that information from one organism to another. And you will see that until today, we primarily use viruses. And that can be linked to the discovery of virus-mediated gene transfer in 1968. So the last is that in order to have human gene therapy, you need to have a way to carry DNA, and that is going to be viruses. We're now moving to 1990 and the very first clinical trials for gene therapy associated with an immune deficiency. So ADA SCID, that is the name of the disease, where a young child is born without the ability to produce a molecule necessary for the immune system. And these children are called bubble babies because they have to live in a bubble because they cannot defend themselves against infection. So I'm going to move to this. So this is a slide describing how gene therapy was imagined in the 1990s. So what you have here on the left is a bacteria carrying a normal human ADA gene. And on the right, you have a virus, a retrovirus that has been genetically altered, changed, so it cannot cause disease. And the idea here is that you take the human normal ADA gene and you put it into the virus. The virus is then used to infect T cells from the patient. For those of you who may not be aware, the human immune system is made up primarily of B cells, T cells, and NK cells. And T cells are very important in the way we protect ourselves against viruses. So you take the T cells, you infect them with this virus that is carrying the normal human ADA gene. And then once these cells are infected with that virus, hopefully they begin to express the normal human gene, and you reimplant this back into the child. And the idea here is that you can correct, hopefully permanently, the genetic defect in the infant, in the child that is lacking ADA. So this was being done in 1990. You will notice the date for 1999 is incredibly significant because this marks the death of Jesse Gelsinger, the first patient who dies from human gene therapy. This happens at the University of Pennsylvania in Philadelphia. It is thought that he died not from the gene therapy, but from the virus that was used to infect him. And this virus triggered an immune response that overwhelmed his body and killed him. And so after Jesse Gelsinger's death, the FDA in the United States paused so many gene therapy trials. And so you notice for the last two dates, the events focus on the European Union. Because after 1999, there is a shift to the European Union for advances in gene therapy. This second timeline looks at blood stem cells. So HSC is hemopoietic stem cells. These are stem cells from the blood. And these are different dates of advances using blood stem cells in gene therapy. So just to illustrate what is done here, here is a image from a paper published in 2013. Again, just immediately after the discovery of CRISPR. So again, this is still pre-CRISPR, before CRISPR. You have a patient. What happens in the patient is you have, in this particular case, either Roma. So, are we back on? Yes. Okay. So what happens is there are two things you can do with a patient. You can remove stem cells from the patient, and so that's one. The more recent is to remove stem cells to reprogram them so that they become pluripotent stem cells, and then to correct the disease gene, and then to take these stem cells and put them back into the patient. And so you have stem cell-based therapy today in order to correct the genetic defect. So this is where we are right now, right? And what you're going to see is these are different viruses that are being proposed to correct many of these other genetic defects. So SCID, this is severe combined immune deficiency. These are all diseases that lead to the absence of the immune system in a patient. And the goal here is to correct the defect with gene therapy. So now we're going to move to the science. So I would like to review some of the science for you. I'm going to begin with the very basics to talk about CRISPR, to understand what CRISPR is all about. So CRISPR is a genome editing technology. So we'll begin with the genome. The genome is all the genetic information of an organism. So here is a human family, and I take a human cell. The human genome can be divided into two parts. The primary part is the nuclear genome. The nuclear genome has about 20,000 genes. The mitochondrial genome, about 40.
Sorry.
Yes.
40.
40.
The nuclear genome is DNA.
Yeah, both of these,
Both?
both are DNA. So both are DNA. So this is the nuclear genome is made up of DNA in linear configurations, which are called chromosomes, and the mitochondrial genome is DNA in a circular structure called, this was the mitochondrial genome. So most of the genetics is in the nucleus, and the nucleus is the control room of the cell. If you take one of the chromosomes, so I don't know if you remember, how many chromosomes does a human being have? Does anyone remember, those of you who are medically, how many chromosomes? ¿Cuánto? 46, 23 from the male, from the mother, from the father, and 23 from the mother. So the total is 46. If you take one of the chromosomes, so if you take one of these chromosomes and you stretch the chromosome, you get the double helix. And the human genome, if you stretch it out, is about 2 meters long, 7 feet. It's very long. And somehow you must put it together so it fits inside a cell And the cell is the size of a period at the end of a sentence. New Times Roman font 12. That period is the largest cell in the human body. What is that cell? Does anyone know? Yes. The egg cell. So the human egg cell, the largest cell, is about the size of a period at the end of a New Times Roman font 12 sentence. Just to give you a sense, and you need to take DNA that is 2 meters long, and you need to put it in, and it cannot be spaghetti. Because if it's spaghetti, you cannot find the information. And so the DNA is wrapped in a very structured way, and you see that here. It's wrapped, and the next 100 years is to try to understand how this is read, and how the information is read, and how the information is used in the cell. Now, if you look at DNA, DNA is often displayed like this. So DNA is made up of 4 different molecules, and each molecule is given a letter, G, A, T, C. And so the order of the letters tells you the information of the DNA. And here you see the information of one gene, the gene that my students and I study. This is the gene. And if you know the code, you will know that ATG is the beginning of the gene, and TAA is the end of a gene. Just like in English, and I think Italian, the beginning of a sentence has a capital letter, and the end of a sentence has a period. The gene also, it begins with ATG, and it ends with TAA. And if you are a molecular biologist, you can read the information of this gene. And my students and I have spent the last 8 years studying this gene. This gene is important because too many copies of this gene, too much expression of this gene leads to cancer of the prostate in humans. So we are trying to understand prostate cancer by understanding what this gene does, and we are now looking for drugs to prevent the gene from working in cancer. And the human genome has 3 billion of these letters. So 3 billion letters. 3 billion, and because we have one from mom and one from dad, it's 6 million total, 6 billion total. Okay, this is CRISPR. So CRISPR is a molecular editing machine. If the genome is like an encyclopedia with 46 volumes, 1 chromosome, 1 volume, then the CRISPR molecular editing machine will allow you to go to page 42 of volume 3, column 2, line 44 from the top, and you change 1 letter. That is the specificity of this machine. It can change a single letter. Now, the machine is made up of 2 parts. You have what is called an RNA, a guide RNA, and then the Cas9 protein. And so the guide RNA, the gRNA, targets the Cas9. It brings the Cas9 to one place in the genome. And then the Cas9 will cut that one place. Now, a lot of people will say, what does it look like? Is it a machine? And they think it looks like a machine like this, like my phone. It's not a machine like my phone. This is what it looks like. So this is a drawing of CRISPR based upon the crystal structure. The crystal structure is what it looks like. So blue is the DNA. The green is the guide RNA. And the yellow is the Cas9. And the Cas9, remember, is what cuts the DNA. And so what we can do is we can design the guide DNA, this green DNA, we can design it so this machine will go to only one place. And it will cut in only one place. So in 3 billion letters, it will cut in one place.
Sorry. So this molecular machine is an enzyme.
Yes. It is. The question is the machine an enzyme? It is. It is a ribonuclease. It's a nuclease, yeah. It cuts RNA and DNA, so it's a nuclease. Okay, so let me show you what happens. So here is the yellow. This is the scissors, and then you have the guide DNA, which is in green. And so what you can do is you design this guide DNA, and it will target to one single spot, and it will cut. Now, when it cuts the DNA, two things can happen. On the left side, if you cut it, and only cut it, the cutting will destroy the information. So if this is a gene that causes cancer, if you cut that gene, the gene will be destroyed. So this is the left side. On the right side, you cut the DNA and you provide a replacement information. And what the cell will do is it will replace the old information with the new information. So the left side you can use to destroy, the right side you can use to change. Questions? Yes.
If you can change only one single letter, what does the palindromic thing?
You can change one letter, you can change 10 letters, you can change as many as you want.
But the palindromic thing, do they have to be actually palindromic?
No.
And why is CRISPR, the P stands for palindromic?
This is not from here, it is from where it came from in bacteria, as you'll see in a minute. The palindrome is not here, the palindrome is in bacteria where it came from. So what you see, you can replace or you can destroy. And sometimes you want to destroy the information, other times you want to replace the information. So this is, if you ask the question, where is CRISPR from? Where did we find CRISPR? CRISPR was found in bacteria. And here it was used in bacteria because bacteria use CRISPR to defend itself from viruses. So on the left side, you have a bacteria that is infected with a virus, and the virus injects its DNA. This DNA is put into the bacteria's own genome in palindromes, which is why it's called the palindromic sequence. But we took it out, and so it's not more palindrome. So the idea is that here, this red virus infected bacteria, the bacteria survive, it remembers the virus's DNA, and then when it is invaded again, what happens is CRISPR attacks the virus's DNA, cuts it, and destroys the virus before the virus can kill the bacteria. So CRISPR naturally evolved as a defense mechanism of bacteria against viruses. But we are using it as a genetic molecular editing machine. So we took it from bacteria and we can put it into any cell and it can do things.
No, sorry. Can we come back?
Yes?
I would understand. I would like to understand. CRISPR, this molecular machine was born to editing.
So
the DNA.
no. So this machine was not born to edit. It was born to cut. We use it to edit.
Okay. And this is the first goal that the second is that can protect.
No. So the machine was found in bacteria. It was found in bacteria because the bacteria used the machine to cut viral DNA. But two scientists, one in France and one in the United States, took the machine from bacteria, changed it a little so that we can use it to edit. But bacteria did not use it to edit. Bacteria used it to cut. We are the ones to take the cutting ability and use it to edit. So you can see here, 2012, Jennifer Doudna, she is a scientist at the University of California in Berkeley. She will probably receive the Nobel Prize either this year or next year in the next couple of years for CRISPR, along with Emmanuel Charpentier from France and someone else. We do not know who that third one is. But in 2012, they were able to show that CRISPR could be used to edit genes. And now, since then, so it has only been 6 years, we have been able to use this CRISPR to edit the genes of many different kinds of animals, including mice, fish, primates, so this is monkeys, and plants, and eventually, as you will see, human beings. So these are pictures of CRISPR-edited animals. These are the real CRISPR-edited animals. On the left here, you have two dogs. One of them is named Hercules. The other one has a Chinese name, so I don't remember it. But these two dogs, beagle dogs, were born in China. The myostatin gene was edited in these dogs, so they are all muscular. You see the muscles of this dog? So these are puppies, but they're muscular because they have one gene, myostatin, edited, and these dogs are being made for the police. And so the idea here is that these dogs will be used by the police. If you land in the United States, they already use these dogs to smell the luggage, to look for food. And so now we want to make the dogs muscular. On the bottom, you have mini piglets. You notice the size of the cup. So the pig is this small. It has been edited so it will be small because it is being sold as a pet pig. Because there are people who want pigs for pets, but the pig is too big. So they made a mini piglet. Again, this was done in China, and I think it's $1,000 to buy a piglet like this, a mini piglet. And then here you have cows that are genetically engineered, so they are resistant to tuberculosis. So these cows have carry a genetically engineered mutation so that they will be resistant to tuberculosis, Mycobacterium tuberculosis. And so these are different animals today that have already been edited using CRISPR in order to do different things. my students want genetically engineered pets. So, you know, the idea now is we can make our pets. You want a big pet, you want a small pet, you want a blue pet, you want a green pet. You can genetically change the gene so that your pet will be a particular color and shape and size. So you can see that this is going to be the revolution of the future. Is there a question in the back? Yes. You have the
one question. You should do it in the first generation and then you don't have to do it anymore, right?
so
Just right.
if these are in the germline, and I'm not sure if these are in the germline. So it is not clear to me if these mutations are in the germline. So the germline is, we will talk about the germline later. The germline is the eggs and the sperm. So if these animals carry the mutation in the germline, then their children will also be genetically modified. I do not know. Now, if you're a company and if you're selling micro piglets, you probably do not want it in the germline. Otherwise, the piglets will produce other piglets for free. So you want it in, you want them to come to you to buy the piglets. So you cannot breed the piglets. Okay, so, but this is, I do not know. I have not read the papers to know the details about the piglets. Okay, now, CRISPR has also been used in mice to correct disease. So I'd like to talk, this is a paper published in 2014. This is a mouse model of human hereditary tyrosinemia. So this is a mutation, a single mutation in the gene called FAH. And the liver, so this mouse is sick. It has a diseased liver, and the liver is not able to metabolize tyrosine. And so the mouse gets sick. And in 2014, they used CRISPR to change the letter of the mouse, of the gene, from a G, an A, so the G is replaced with an A. And what happens is they repair the liver, and the mouse is cured of the disease tyrosinemia. So this is a model for human gene therapy, because not every cell in the liver was changed. Only some of the cells were changed, but enough of the cells were changed that the disease was cured. So this is 2014. Now, this is another proposed use of CRISPR. This is called gene drives. So this is the way that you can use CRISPR to edit the genome of an entire animal population. And this is being used today for primarily controlling insects. So what happens here is the red insect is carrying CRISPR as well as the gene that you wanted to carry. In normal inheritance, in normal hereditary, when one red fly mates with another fly, blue, there is a 50% chance that the children will receive the red gene. Using CRISPR, what we can do is we can convert, so this is the red gene. Once the red gene is present with the blue gene, the red gene will change the blue gene into red, so that you then, even though you only inherited one red gene, all of your genes become red. And so what happens is that all of the genes are eventually replaced by the CRISPR gene. Now you should know that if you are able to introduce the CRISPR into 1 in 1,000 animals, all you have to do is wait 12 to 15 generations before every animal has that gene. It would be true as well, but 12 to 15 generations would be a long time for us. It would take 500 years. But for insects, 1 or 2 years. Now it's interesting because it's already been tried. So this is an experiment by a company from the United Kingdom called Oxitec. They went to the Cayman Islands in 2009. Now, the reason why Cayman Islands is because it's an island, so the mosquitoes couldn't go anywhere. What they did is they released 400,000 mosquitoes every week. 400,000. And they noticed that over the generations, you had 88% reduction of eggs and 62% reduction of the virus-carrying mosquito. And so this is an attempt to alter the ecology of the environment to decrease infectious mosquitoes. Now, CRISPR can also be used to edit human genomes. And there are 2 ways of doing this. This is in vivo on the top, where you use a virus, and the virus will carry CRISPR into the patient. This is in vivo editing. And in the bottom, you have ex vivo editing, where you take the cells from the patient, remove the cells, edit the cells in the laboratory, and then return the cells back into the patient. So the top in vivo, the editing happens inside the patient, in vivo, and the bottom, it happens outside the patient, ex vivo. And we will talk about some of the different technologies used, specific technologies, in a few minutes. I would like to end this section by discussing this paper, Hongma et al. Published last year, which is the first use of CRISPR to edit and correct a defect in human embryos. So this was done in the lab of Shukrat Mitalipov in Oregon. Oregon is one of the states in the western part of the United States. It's north of California. The capital of Oregon is Portland. So in Portland, Oregon, in the laboratory of Shukrat Mitalipov, they were able to CRISPR human embryos that were obtained using IVF. This is just the data, 58 IVF embryos with a mutation in MBYC3. MBYC3 is a gene that is important for muscles in your heart. And these embryos carried a mutation that would have impaired heart function. 75% of the embryos were correctly edited. I have to point out that since then, so in the last 8 months, there have been criticisms of this experiment. I'm not going into the details now. There are criticisms about how this experiment was actually interpreted. Okay, now we move to industry, and we have a break after the industry. So there are 3 major companies in the United States that deal with CRISPR, and they're all located in Boston. And I live in Providence, which is 1 hour by train from Boston. And so last month I was invited to the companies. They wanted me to visit their labs, and I spent one whole day visiting the labs. And they each of them took turns giving me a presentation on what they did. And the reason why these companies are doing it is because these companies would like to educate the public, including the Catholic Church, on CRISPR and what they are doing, because they want to make sure that their industry listens to people's concerns. And I was at a conference in the Vatican 2 months ago, and that's where I met them and they said, "Oh, you live in Providence, come visit." So I had a wonderful visit. They gave me lunch. And so what I asked them is I asked them for the presentations that they gave me when I was there, and I asked permission to share those presentations with you, so you have an understanding of what two of these companies, the third company, the person who I talked to was on vacation, so he couldn't send it to me, but I will present two of the companies to you so that you understand the technology behind the industrial CRISPR project. So the first company is called Editas Medicine, and I thank Vic Meyer. His name is at the bottom. So Vic Meyer is the Chief Scientific Officer of Editas. Now, I will, when I give you the PDF, I will not include these slides because these slides belong to the company. So what I will do is I will give you my slides, but not their slides, just so you know that. They will be missing from the PDF version that I will send you. But I wanted to share some of these slides. So to give you a sense of the company is 4 years old. It is based in Cambridge, Massachusetts, which is across the river from Boston. It's a suburb of Boston. And Cambridge is famous because of two universities in Cambridge. One is Harvard, and the other one is MIT. And these companies are all around MIT because MIT is considered the hub of biotech in the United States. And this year, just a few months ago, they opened up a new office in Boulder, Colorado, in the central part of the United States. They have 125 employees, and they are publicly traded in the United States on the New York Stock Exchange. And I'm going to now talk to you about some of their technology that they are using in order to develop medicines for commercial use. So if you ask, what is the goal of the companies in 2022? So 4 years from now, what are their goals? These are their goals. They want to have 3 drugs, an early stage clinical trials. This is their goal in 4 years. They will have 2 drugs in late stage clinical trials and 1 drug that is being used actively in medicine. So this is a very, very ambitious platform, but the individuals I met from this company are very enthusiastic. They think that they will be able to meet this goal. So these are the diseases that are being targeted by the company. I'm going to focus primarily on the ocular diseases. They want to develop CRISPR for the eye. Now, the reason why they're going to develop CRISPR for the eye is, one, the eye is easy to get to. You can easily access the eye. The second is that the eye is immunoprivileged. So the eye is separate from the immune system of most of your body. So the eye, if you inject things in your eye, it doesn't go to the rest of your body as easily. And there are diseases of the eye that cause blindness that are due to genetic defects. So I'm going to focus on the ocular diseases. And the disease that they are focusing on now is called LCA10, Leber Congenital Amaurosis Type 10. So just to give you a background on this disease, which they are developing technologies for. So what happens is that the photoreceptor, which is the molecule in your eye that allows you to see, slowly dies when the patient is a child. So the patient is not born blind, but over time the patient loses sight as more and more of his photoreceptor cells die in the retina of his eye. There are 2,000 to 5,000 patients in the West, United States, in Europe. And one of the reasons I asked them, I said, why do you have to put this? You have to say, how many people will buy the drug? So they're going to target diseases where there are enough people who will be able to have the insurance to cover the costs of the drug. And they want to submit a request for an IND, is a request for a clinical trial this year to the FDA. So what I would like to do now is to review some of the data that they shared with me with regards to their technology. So this is the photoreceptor cell. The photoreceptor cell is present in your eye. It's present in the retina of the eye. It allows you to see. And these outer segments include the molecules that receive light. Patients with this disease lack this gene, the gene for a protein called CEP290. And because they do not have CEP290, the photoreceptor dies. And slowly over time, they become blind. Now, one of the things that you need to know is that patients with this disease still have photoreceptors. Their experiments have also shown you only need 10% of your photoreceptors in order to see something. So if you have a child who is losing photoreceptors, you could, in theory, genetically engineer the remaining photoreceptors so he can still see. Even if he has 10%, 90% die, but if he has 10%, he will still be able to see, which is why this company is targeting this disease. Now, just to get so you understand how to use CRISPR, and I'm going to apologize, I'm going to become very scientific here. For those of you who are not scientists, you may not miss this. So the disease comes from a splicing defect. Splicing. So in order for you to get a correct gene, you must join exon 26 and exon 27. You must join them together, and you must remove everything in between, which is called an intron. This is called splicing. Patients with this disease cannot splice this intron, so they cannot remove this red X. So what the company is doing is that the company would like to use CRISPR to remove this defect so that you can correct the splicing and get normal CEP290. And that is their goal, to correct CEP290. This is the CRISPR machine. It was used, it is put into a virus, an adenovirus-associated virus, and it is injected into the retina of the eye. Remember, you only need to correct 10% of the photoreceptors in order to preserve sight. Now, this is a summary of the studies from non-human primates. So they did studies with monkeys, and they were able to show that the injection was tolerated well in the eye of the monkey. The eye did not respond negatively to the injection. I did not show the data, but it was able to correct the genetic defect in the eye, and you do not have an immune response to the injection. So it looks good. And the next thing is to apply for FDA approval for use in humans.
We have to buy the stock of this company.
There are people, lots of people are buying stock in this company. The company has very high stock value. All of these companies, it's amazing. In 3 or 4 years, their stock has increased tremendously because these companies are thought to be the cutting edge of medicine. If these work, then these companies will be incredibly valuable in 5 years.
How much would a treatment cost? How much are they charging?
So right now there's only clinical trials. So they do not have a product but just to give you a sense, right? There are products for cancer treatment using for those of you who are familiar with CAR T or checkpoint inhibitors. These companies will pay, you have to pay $10,000 per month for the rest of your life. But insurance will pay for it. In the United States. One of my friends actually, his insurance pays $10,000 a month for his treatment, and he will have to use it for the rest of his life. He's 45. So you see, there's a lot of money. There's a lot of money if these treatments work. All right, so before our break, I would like to give you the second company, Intellia Therapeutics, also in Cambridge, and I thank Tom Barnes. Tom Barnes is also the chief scientist, and he gave me, these are his slides, a few of his slides. So this is Intellia. Now, it's interesting because Editas and Intellia and CRISPR, those are the three companies. They're competing with each other, but they have different diseases, so they're not overlapping. Question. I do not know which one she's an editor. I do not know. It's all confusing to me. They're all trying to make money. And honestly, as a scientist, it troubles me that scientists are now driven primarily by money. You know? Yeah, but she has a company. So there's a little bit of me that is concerned. I have avowed poverty, so there will never be a company for me. And yet, there's a part of me as a scientist that is concerned that when scientists are motivated by money rather than by truth, it becomes more difficult to regulate science. And the desire for knowledge might be in some way distorted. I'm not sure if that will happen, but I am concerned if that will happen. So let me just move to Intellia. So Intellia's value proposition. So what is special about Intellia is that they use not viruses, but lipid nanoparticles. So this is a virus, this is the size 20 nanometers. They use lipid nanoparticles that they will inject directly into the patient. The lipid particle will deliver the CRISPR machine, but then it will eventually be destroyed. And so nothing remains. And the concern if you put a virus is that the virus does things you cannot control. The lipid nanoparticle is much less so. And the idea here is that these are all the things they were able to do. They are targeting diseases of the liver, where they will, not of the eye, of the liver, where they will inject lipid nanoparticles directly into the liver and allow the liver to absorb these lipid nanoparticles and therefore be genetically edited. Now, one of the questions that I'm often asked is, how do you know if CRISPR will not cut anywhere else in the genome? Because CRISPR cuts. And so how do you know it will cut here and not there? But it turns out, so one of the things I discovered is that Intellia asked this question: how often does DNA get cut in any cell? No CRISPR. And it turned out that there are thousands and thousands of cuts that normally happen right now in your cells. So if we are worried, I have CRISPR, it's going to cut once, and I'm worried it's going to cut somewhere else. There is cutting happening normally in your and my cells, because you have a spaghetti that is 2 meters long, and of course it's going to be cut. So one of the things most people don't realize is that you have cutting normally in human cells. And so the question is, if you add CRISPR, how much more will you add to the normal cutting that happens? If there are 10,000 cuts, if you add one more cut, is that a difference? This is what they were telling, and I didn't know that. I did not know this. I don't think many people know this. They had to go and study this. They had to ask how many double-strand breaks. So a double-strand break is a cut. How many normal cuts happen? Thousands happen every day in every cell in your body. It's normal. Question. So your question is CRISPR reversible? Well, you just have to send another CRISPR in. Okay, so that's the idea. You send another CRISPR in. And one of the things that's really interesting is you notice everyone is worried about how specific will it cut. And one of the things that the companies were able to show me is that they were able to go through thousands of guide RNA. Remember the guide RNA that's supposed to target CRISPR? What they do is they try thousands and thousands until they find one that is so precise it will cut only once. And so, for example, here are many, many, many different kinds of target guide DNAs, and they just select. So if for the patients with CEP290, they will find the perfect, ideal guide RNA that has very low background, will only cut once. And they're doing this now over and over again. It's not random. There's a question in the back.
My knowledge of CRISPR is very limited and very colloquial, but my understanding from reading the media was that in embryonic experiments that there was potential for mosaic embryos. That is CRISPR.
Yes, so those were the experiments done last year in Oregon. So if you're doing it with embryos, there's going to be mosaics. If you do it with patients, there's also going to be mosaics. So a mosaic is half normal, half corrected, or some combination of the two. And even with the correcting of the livers of patients, you don't need to correct every cell in the liver. You just need to correct enough cells in the liver so that the liver will be able to function normally. And the liver And in the eye, you only need 10%. In the liver, I understand it's something 20 to 30%. And if you can correct 20 to 30%, it doesn't matter what happens to the rest of the liver. Well, with embryos, it's portrayed negatively because we don't want half of you to be sick and half of you not. But in corrections for adult patients, the mosaic is not as much of a problem. But for embryos, I have problems with embryonic experiments to begin with, let alone whether or not they're mosaic, because they killed the embryos. But that's a different story. So this is the gene, this is the disease that this company is trying to correct. It is a transthyretin amyloidosis, ATTR, and they are doing this primarily by injecting their nanolipid particles into the liver, carrying CRISPR to edit. Now you notice there are 50,000 patients with ATTR worldwide, and it's usually fatal within 2 to 15 years of age. And the idea is that we would correct it as soon as the child is born. So 2 to 15 years, typically fatal. And the idea is to try to correct it in the newborn baby liver before the child can die. And this is just experiments in the mouse. And in the mouse, what they were able to show is that they can edit 70% of the cells in the mouse. And this is actually, this is mosaic, it's only 70%, but they were able to reduce the toxic protein by 97%. So the idea here is that in patients, this would be incredibly curative and therapeutic in patients if this is what we could do with babies, as we can do with mice. And they also showed me this. This is the first time they were able to do editing in the brain of the mouse. So this is central nervous system editing in the brain of the mouse using their lipid nanoparticles to cross the blood-brain barrier and to inject directly into the brain. So the idea here on the left is you have the mouse, on the right you have non-human primates. And again, the idea is that we would eventually try to correct genetic defects in the brains of patients. Finally, this is their ex vivo program. They're attempting to correct acute myeloid leukemia. And the idea here is that this is very low survival rate, 5-year overall survival of less than 30%. And the idea is that we will correct the genetic defect in AML in order to cure patients of this cancer. So let's take a break. Yes, question from the front.
This CRISPR editing tool can switch also the part of DNA or only cut?
We'll cut and replace.
But not switch.
I mean, you replace with something that you put in. So it switches in. Okay. All right, let's take a 5-minute break before moving to the ethics of this technology.
How many worldwide, how many trials? I mean, these companies are just a sample they give. I'm sure there are a lot of other trials, or I'm not sure if it's true. So how explosive it is, just to have an idea.
China a lot because it's unregulated completely. In fact, when you talk to these companies, their concern is that in China, you have so many unregulated experiments that the danger is one patient will die and completely soil the entire industry. But in the United States, there's only 3. These are the big 3. There's maybe 1 more. Okay. And then in Europe, I think there may be 1. So there's not that. I mean, there's a lot of experiment. There are many, many labs doing this experiment, but there are only a handful of companies. But these companies are valued highly right now on the stock exchange. Thank you very much. Let's take a break. Presentation, I would like to focus on the ethics. And I'd like to begin with a couple of distinctions to distinguish different things with CRISPR. So we already have gone ex vivo versus in vivo. Ex vivo is the genetic editing of the cells happens outside the patient's body, while in vivo gene editing happens inside the patient's body. The second distinction is somatic versus germline. Somatic cells are all the cells in your body except for the reproductive cells. So most of the CRISPR gene editing can happen to correct mutations in your lungs, in your liver, in your pancreas, in your kidney. That is somatic gene therapy. If we change the genes in your sperm and eggs, we are doing germline gene therapy. And so there is a big distinction between changing the patient or changing the patient and his children. And finally, there is this distinction we discussed on Monday, therapy versus non-therapy, where if you remember, therapy cures, prevents, or delays illness and sickness. Non-therapy seeks to increase personal or social opportunity. So these are the differences. The first thing I would like to discuss now for the ethics is we will begin, before we deal with human ethics, I would like to talk about the ecological ethics. This is primarily the ethics of gene drives. And we are dealing here with active Zika transmission. So if you remember Zika appeared several years ago. It is a mosquito-borne disease. And it is particularly devastating when pregnant women are infected with Zika because it leads to congenital defects, the lack of formation of the brain, microcephaly of the child. So these are many of the countries that are thought at this time to have active Zika transmission. This is the mosquito that carries Zika, Aedes aegypti, also carries dengue, yellow fever, and malaria. Now, if you look, this top here is a image taken from the website of the Oxitec company. The Oxitec company is the company that is using CRISPR to try to lower mosquito populations. If you look, it says the female mosquito is the most dangerous animal in the world. Because every year mosquitoes spread disease that kill 1 million people every year, and it debilitates hundreds of millions more. So in their view, the female, not the male, the female mosquito is the more dangerous than a tiger or a shark or an alligator is the female mosquito because it kills more people than these other animals. Now, you should know that there are about 3,500 species of mosquito. They want to target one species of the 3,500 mosquito kinds. And this is Aedes aegypti. And so the question is, so just think about how much is spent for dengue. Every year, $380 billion is spent on dengue, and Zika, $3.5 billion is spent on Zika. So there's enormous amount of money spent trying to prevent, to treat these and prevent these diseases. the question is, if we use CRISPR gene drives to eradicate one species of mosquito, is that good or evil? Now, we have already removed smallpox. So smallpox has been eradicated. The question now that I would like to raise here is, if Oxitec wishes to eradicate one species of mosquito out of 3,500 species of mosquitoes, is that good or evil? Okay, so I want you to think about that. That's ecological ethics. We can talk about that at the end of the lecture. So now we move to the ethics of human gene editing. And I will focus on two documents of the Catholic Church. The first is Dignitas Personae, and the second is Humane Vitae. And both of these documents have something to say about CRISPR, the ethics of CRISPR. So the first is Dignitas Personae. The first line of Dignitas Personae, which is the document from the Congregation for the Doctrine of the Faith, the main guiding moral principle of Catholic bioethics is the following: the dignity of a person must be recognized in every human being from conception to natural death. And I gave this a short version of this presentation last Friday, so a few of you have seen a few of these slides. Now, it's not just in the Catholic Church, because the first sentence of the German Constitution, the Grundgesetz, also begins, human dignity shall be inviolable. So human dignity is at the heart, at the center of the Catholic worldview, as well as the political worldview of at least the German Federal Republic. But the question is, what is human dignity? And so we need to start with this discussion. And human dignity is an answer to this question: how much are you worth? And when I teach this in my class, I ask my students, how much are you worth? And if I ask your mother, how much are you worth? My students will say, Father, I am priceless. And that is true. And so human dignity is an explanation for why you are priceless. So the example I use in my class is an iPhone 7. If I say, how much is this iPhone worth? There are two numbers associated with the value of this iPhone: the intrinsic value and the extrinsic value. So the intrinsic value, if I take this iPhone and I take it apart and I ask how much is each part worth, it comes down to $219.80. This is called the tear down cost. If I ask how much is this worth on Amazon or on Apple.com, the suggested retail price is $649. So you have two numbers: the intrinsic value, how much the parts are worth, and the extrinsic value, which is the worth of this phone on the open market. Now, one of the things that you should know is that the intrinsic value of the phone never changes. Once this phone is a phone, it costs $219.80, and it will cost $219.80 until you pull it apart. But the extrinsic value of this phone will change. If you buy it on Amazon, if you buy it in Roma versus in New York, the price will be different. It will depend on so many social and environmental factors, because this retail price is variable. So the Catholic moral tradition and many other faith-based traditions also claim that human beings have two worths, an intrinsic and an extrinsic worth. So the intrinsic dignity is the inherent worth of the human person. You and I are priceless. The extrinsic dignity is the social worth of the human person. It's how much other people think we are worth. And usually this is associated with your salary. So if you are worth more, your salary is higher. And the net worth, for example, of Bezos, who's the owner of Amazon, is now like billions and billions and billions of dollars. That's his net worth. And yet, he is of equal intrinsic worth to any of us, even though none of us, I don't know, but I don't think so, any of us here is a billionaire. Okay? So there's the 0.1% and everyone else, and we belong to the everyone else. So there's two values, the extrinsic value and the intrinsic value. And the same thing with celebrity or high office. So I understand recently Justin Bieber got engaged. I just found this out. My students tell me this, okay? Justin Bieber got, and one of my students is upset. She wanted him for herself. But, so now he has celebrity. So he is worth more. But that is his extrinsic worth, not his intrinsic worth. Now, you notice the intrinsic dignity is how much you and I are worth simply because we're human, nothing else, just because we're human. And you do not earn it, and it is not given. It is acknowledged and recognized. Other people say, you have intrinsic dignity. Notice intrinsic dignity is absolute. You cannot have 50% intrinsic dignity. It is 100% or 0%. Because this value is worth depends upon you as being human. If you're human, you have 100%. If you're not human, you're 0%. So there is no such thing as partial intrinsic dignity. And for Christians, the intrinsic dignity is grounded upon a theological claim that we are the imago Dei. We are made in the image and likeness of God. And this is the anthropology that you are learning about this week if you've not heard it before here at the Regina. And so for Christian and Catholic bioethics, everything that must be done to protect, preserve, and advance both the intrinsic and extrinsic human dignity of the human being. Especially the intrinsic dignity of the human being. And if you do not do this, if you do not recognize and acknowledge the intrinsic dignity of the human person, then this action is immoral and should therefore be rejected and not pursued. Yes.
I'm sorry, I interrupt. But people who don't believe in God,
Yes.
have any God, or
Well, this is a question. So Father Joseph and I are having this conversation over lunch. The question is the following. If you do not believe in God, can you affirm the intrinsic dignity of the human person who is priceless? I do not think so. I do not think so. Exactly. If you're an atheist, it is very hard to show that you are worth more than a dog.
The criminals, the people who are doing only evil actions against the other.
Why should I not kill them if I can kill a dog? Again, this is where the debate is. This is a debate. I've been in a debate with some other people in the United States. So I have a colleague who says, isn't it obvious that the human person, even without God, is more valuable than a pig? And I say, why? He says, well, human beings can make music. Human beings have culture. Human beings have civilization. And I say, why is that more valuable? Ants can make an ant colony. Why is a city more valuable than an ant colony? Now, usually you will say, well, because it involves things that are intellectual and spiritual. So if you think that spiritual things are more valuable than material things, then of course you will say that music and reason and will are better. But if you don't believe in spiritual things, if everything is just material, and the standard by which you decide what is more valuable, as you will see in a minute, becomes biological. And once it is biological, then there is no reason to think that being intelligent and being beautiful is more valuable than being powerful. So this is, and I'm having, this is a constant debate. And I grew up in Thailand, which is a Buddhist country. And one of the things in a Buddhist country is it's not clear why a human being is more valuable than anything else. As my ex-girlfriend said a long time ago, if you can be a mosquito in the next life, then the mosquito is just as valuable as you. And it's a very interesting thing. And she would not kill a mosquito because that could be her grandmother. But you see, the value depends upon your worldview. And I think in the West, we are so deeply Christian, even though the culture is now post-Christian, the assumption that the human intellect and the human will, going back to Socrates, Plato, and Aristotle. Aristotle said, intellect is a greater power than anything else. And I go, why? I'm a biologist, and as we will see in a minute, there is some reasons why I will choose other things than being human. But we'll go back to that in a minute. So, for example, if you are Ruth Macklin will say dignity is a useless concept. This is a very, very famous paper in 2003. She said, dignity is useless. Dignity should be autonomy, the ability to choose for myself. And she's coming, of course, from Kant. And this is where I think the Enlightenment tried to replace dignity. The Enlightenment attempted to ground the Imago Dei in something other than a theological claim. And I believe it has failed. I do not think that the Enlightenment has been able to argue for the exceptionalism of the human person outside of a theological framework. I think it's failed, which is why we are so confused today. So I think that the Enlightenment, which has attempted to justify the exceptionalism of the human person apart from God, I think the Enlightenment has failed. I do not think that an atheist or a secular person can properly justify the exceptionalism of the human organism over any of the other organisms on this planet. Each organism is uniquely adapted to a particular environment. And in certain environments, humans are great. In other environments, humans are terrible. And so depending upon the environment, the value changes. And this is something I think that Ruth Macklin and the others have failed to understand. So she says autonomy is better than dignity. And Steven Pinker will say the stupidity of dignity in 2008, that dignity is stupid. Everything should be autonomy. It should be freedom. And so for some secular bioethicists, human dignity means no more than respect for personal autonomy. But the question I ask is this: why is autonomy worthy of respect? Why is autonomy valuable? Autonomy is the freedom to choose. In order for you to have autonomy, you must have intellect, which is knowledge, and you must have will, which is choice. From a biological perspective, intellect and will are evolutionary adaptations. And there are many adaptations in the biological world. And one is not better than the other, absolutely. It depends upon the environment. I'll give you an example. I say, if tomorrow you wake up and you find yourself in the middle of the polar tundra in the Arctic, would you choose to be an intelligent human being, naked in the snow, singing music? Or would you prefer to be a polar bear? Which is more valuable? And I will say that polar bears are more, it would be more valuable for me to be a polar bear if I found myself in the Arctic than to be a naked human being reciting Dante. It doesn't matter if there is Dante or if there is music, I will be dead. The same thing happens in the reverse. If you want to be in Roma, you want to be a human. You do not want to be a polar bear, because if you are a polar bear, you will be a dead polar bear in the Piazza Navona, because we will shoot you there. So from a biological perspective, each species is valuable in a specific environment. And it is hard to argue from a secular perspective why the human species is inherently more valuable always, at all times, in every environment. And I think, and I know it's controversial. Now, I, of course, think that the human being is valuable and the human being is priceless. But I think that the justification for that is theological and not philosophical. And when I argue with my atheist friends, and my atheist friends want the human being to be superior to all others, and they want men and women to be equal, I say you have to be Christian to say that. And that is what I'm saying. And if you do not say that, you're incoherent. There is no reason for you. If I ask, why are men and women equal?
Because we are all human beings entitled to the same rights.
Who? That in Thailand, that is not true. So the question, so the response is, we are human beings. Why are men and women equal? They are not equal. We're not physically equal. This is why there's the Me Too movement. Women have to be protected against men, because men will be predators against women. Exactly. So this is why, if there is nothing we can identify other than intrinsic dignity that you can argue that all men and women have equally. In fact, I will argue this is why so many societies are inherently patriarchal, because the patriarchal structure is biologically based. If you look at non-human primates outside bonobos, the male primate dominates over the female. So the biological basis, the biological state of the primate is primarily male dominance over the female, suggesting non-equality. And the only reason why we argue that males and females are equal is actually precisely because of a deeply, deeply Christian claim that we are all equally sons and daughters of the Father, made in the image and likeness of God. Growing up in a non-Christian culture, I will tell you a story. A long time ago, before I knew I was going to be a priest, I was dating this girl. She was Buddhist. And she said, when we are married, she will choose my minor wife. I said, what minor wife? And she said, well, you are a man. You are going to go out to a brothel. So instead of you going to prostitutes, we are going to pick a minor wife. She will live downstairs and I will live with you upstairs. And I said, no. I said, you and I are equal. There's only one for me. She says, that's not true. She says, look around. There's guys cannot control themselves. It was so shocking for me. I realized I am inherently Christian. I assumed that men and women are intrinsically equal. But in a non-Christian culture like the Buddhist culture in Bangkok, the assumption, looking at animals, looking around, is that men and women are not equal, that men are more promiscuous than women, and that this is something that she has to accommodate.
But that we're not equal doesn't mean that we're not worth the same.
Well, if we're not equal, we're not worth the same. Well, I ask you, is the iPhone 7 and the iPhone 10, are they worth the same? Why not? Because they're not equal. Exactly. But if I have... Exactly. So the question is whether or not the equality of everyone is a cultural claim. Is it a cultural claim? Is it just simply cultural? And this is where it's a deeply deep, this is where our postmodern, post-Christian secular culture is having this incredible debate, right? If two iPhones should be worth the same, for the most part, i7s, but an i7 and an i10 will exactly, but what's so special about being human?
They think human men and women are the same. And secondly, Aristotle didn't think that women are the same men.
Yeah. So exactly. So first of all, an atheist society that claims that men and women are equal.
Yeah, you don't need to be Christian to think this.
Well, actually, this is.
I think it's very offensive to say this to people like me, to people like her, probably.
All I'm asking is this: why? I am saying that in the West, the atheist, the secular claim that men and women and all human are all equal.
We're equal.
Yes, that is actually
all the culture that think this.
That is not true.
It is
that is not true. I grew up in a culture that is not. It is not true. It is not true. Buddhist cultures do not think that all people are equal. It is not true. I mean, it is a profoundly Christian. If you go to India, if you go to Thailand, it is profoundly cultural that there is not a belief that every. Oh wait. Yes, but the thing is, what is so striking is, one of the things that I would like to point out is that Western European culture. No. I'm saying that Western culture, whether Western culture realizes it or not, is indebted. It is post-Christian. It is after Christian. It is a post-Christian culture. If you go to cultures, I'm sorry?
The Enlightenment came before.
All I'm saying is this. I am very confused with your account because I grew up in a non-Christian culture that has never been Christian, ever. So if you go to a Buddhist culture, Thailand has never had a Christian culture. And in a non-Christian culture, it is not self-evident that every human being is the same. It is not self-evident that men and women are equal. All I'm saying is that in cultures where Christianity has historically been present, you have this claim. And the question I would like to ask my friends at MIT is, how do you justify the claim that men and women are equal that all human beings are equal in a secular way. Even Aristotle, who was secular, was struggling. So let's wait. Let me finish this presentation and we will jump back to this, all right? So that we will just finish it up. So what I'm going to do now is, in contrast to autonomy, dignity has a lot of explanatory power. So you see, this is what I'm saying. Dignity explains why we are equal. Dignity explains why we are more valuable than plants or more valuable than cows. You know, I mean, in India, cows are more valuable than humans because cows have sacred value in the way that humans do not. And so I think we fail to appreciate how Christianity has influenced the way we look at the world. So for faith-based bioethics, dignity is the ground for autonomy. As such, dignity concerns can limit and constrain autonomy. So what I'm going to do now is I'm going to just finish up with Humane Vitae. So Humane Vitae, which is 50 years old this year, Humane Vitae deals with how it has to do primarily with human procreation. And this is called the inseparability principle. And I do not have an opportunity to go into the details now. But in the order of nature, procreation is intrinsically linked to marital union. So this is Pope Paul VI, who will be made a saint in October. The idea here is that this is the Catholic Church's principle that undergirds the way that she thinks about artificial reproductive technologies. And so what I'm going to do now is, in order to talk about CRISPR, we need to talk about how CRISPR looks at both the inseparability principle as well as the preservation of human dignity. So these are the guidelines. So the first thing, these are the 4 things we need to think about: the safety of persons, the commodification of persons, the marginalization of persons, and just access for all persons. So this is the situation now that we're going to talk about. So the first is the safety of persons. What I hope that I was able to show you, especially the companies that are dealing with CRISPR, is that they are working very hard to make CRISPR safe, to decrease off-target mutations. These are changes cutting in somewhere else other than where you want to target. And then chimeric tissues. Chimeric tissues are tissues that are half edited and half non-edited. And the question here is, can we collect enough of the cells in a tissue in order to get it working again? Second thing is commodification of person. So commodification in English is to treat a person like an object or a product, something that is made. Exactly. And so here, if parents are allowed to design their children to choose all of the characteristics of their children, is this not treating the children like products of the parents? So that's a concern here. Another concern has to deal with IVF and artificial reproductive technologies. And I have no opportunity at this time to explain how the artificial reproductive technologies and IVF commodify a person to reduce a person to an object. And what happens here is persons are made in the image of another rather than made in the image and likeness of God. So if you look at the current model for CRISPR for humans, it involves IVF. And from a Catholic perspective, this would be difficult to accept because it would commodify, objectify the person. And so what sort of other options are available? These are 2. The first is we take cells from the testicles of the father and genetically edit the stem cells from the testicles and replace them back into the father. So we can now, the father, the male can reproduce children like he normally would, but now his sperm have been genetically edited. So this is for the male. And for the female, there are experiments in the Tully lab at Harvard that suggest that the ovaries of the female contain stem cells, and that these stem cells can be edited, return to the ovary, and the ovary would then give rise to genetically altered pups. So in this particular case, what happens is you have stem cells that are genetically edited, so they are green in color. So they have GFP, they turn green, and you put them back into the mouse and the rat have now green puppies, green pups. So the little baby mice are green and the baby rats are green because the ovarian cells that were genetically altered were able to turn into eggs, which were then fertilized with sperm. So this is the idea, is that if you wanted to change and genetically edit the germline, you would change the sperm and the egg by changing the stem cells in the testicles and in the ovaries. The final concern is the marginalization of persons. So the difficulty we have is that if we design our babies, who decides what is ideal for the children? So I have students who want their children to be white, to be blonde, and to have blue eyes. And the danger is that this sort of genetic engineering will make the social divisions in our cultures worse. And it will, the eugenic temptation, eugenic means to make the population better. And the idea is that who decides what is better, especially since what is better is often associated with the people who are powerful and the people who are rich. And so the danger here is that you are going to exacerbate, make worse the divisions in our society. And finally, especially in the pontificate of Francis, we have to ask the question, will CRISPR be available to the poor? Or will CRISPR only be available to the rich? And right now, if CRISPR costs $10,000 or $20,000, and you have to have very good health insurance, then only the rich and those who are able would be able to have this. And so when I go to the companies, I tell the companies, how will you make sure that the drugs that you develop will be accessible to the poor? And when I have this meeting with them, for example, one of the things we've talked about is, can we set up a center in Kenya, for example, so the technology that is based on CRISPR requires one factory in order to genetically edit these cells. And the question is, and they were saying, well, what happens if we sell our machines to the Catholic Church, who will build one hospital in Kenya that will give the African people, especially those who are poor, access to these machines? So we're having, it's corporate social responsibility. I'm challenging people to challenge, the Catholic Church challenges these companies to imagine ways where they can take their products and make them much more available to those who are poor. So this is the concluding slide. I'm going to propose that somatic cell modification, so changing the cells of the body for therapeutic reasons, are probably good. Germline modifications for therapeutic reasons, for changing the germline so that children do not get born sick, that could possibly be morally justified, again, as long as everything is safe. But germline modifications for non-therapeutic enhancement, so this is designing babies so that your baby looks like you, or your baby is tall, or your baby is beautiful, those would probably not be ethically justifiable. So I just like the last couple slides to thank, this is my laboratory, these are my students. My students talk to me all the time about CRISPR. Half of them believe in God, the other half don't necessarily, so it's a wonderful conversation because especially the ones who don't believe in God, they ask me the hard questions. They're always asking, why? Why is this? And then I ask them back, why do you think this? And we have many, many conversations, usually late at night. And they help me to think about what it means to be a human being. So this is my laboratory. And then this is the meeting I had on June 13th, so about a month ago. These were the representatives of the 3 companies that we met in Boston, in Cambridge, in order for them, they spent the whole day, they took me on a tour of their labs, and they gave me PowerPoint presentations on their work in order for me to understand better what they are trying to do. This is the money that my laboratory receives for funding for the research labs and the bioethics that I do. And also, one of the things I do is evolution, because there are many Catholics who do not know if evolution is true. And so this is a website that my Dominican brothers and I have put together, themysticevolution.org, where we use the thought of St. Thomas Aquinas, the great Dominican theologian and philosopher from the 13th century, in order to help us today in the 21st century to think about evolutionary thought. Thank you very much. All right, back to questions. All right.
There's a problem here with the break time. So,
okay,
how do we do this?
what time is the, so maybe we had a break, so let's just go for 5 or 10 minutes of questions, then break, and then, okay. So from the
I'll
front,
be the time.
you'll be the time. Father Joseph will be the timekeeper.
The question is, if you think, is there any difference in your opinion from gene editing or gene manipulation to eugenics? Is it the same? Is it different? And if it's the same, is it necessarily bad?
So eugenics is an attitude. Eugenics is an attitude to improve the human race. Gene editing is a method. So gene editing can be used for eugenics. Gene editing can be used for therapy. And if it's used for therapy, I think that's good. If it's used for eugenics, I think it depends, right? I mean, if you think that the human race should all be 6 foot tall, blonde, blue-eyed, and we are going to remove everyone else, that's eugenics, and that's bad. The Nazis tried that. Next question.
Ecco, io pongo la domanda in italiano, non so se Jay deve fare qualche... Ok, io vorrei tornare sul concetto di dignità, se... Immaginavo. Allora, io sono d'accordo con lei che è estremamente più facile giungere al riconoscimento del valore intrinseco con un approccio di fede cristiano, no? La cultura cristiana sicuramente è giunta come dire, a facilitare questo riconoscimento, non c'è dubbio. Però io non escluderei, come mi sembra ha fatto lei, la possibilità anche per gli atei e le altre persone di altre religioni a giungere alla stessa conclusione. Per quale motivo? Innanzitutto perché anche gli atei e i buddisti sono creati a immagine e somiglianza di Dio. E allora questo che cosa significa? Che anche per loro, attraverso la ragione filosofica, però esercitata in maniera onesta, si può arrivare al riconoscimento del valore intrinseco della persona umana.
So you have to translate that all, because I don't understand Italian.
Basically, what we're going to talk about at lunch.
Okay.
So basically, she was saying that the fact that even those who are atheists or Buddhists, they're created in the image and likeness of God, so they should be able to deduce some way through reasoning, okay, that there is an intrinsic dignity. So philosophically, she thinks that it's possible through reflection, self-reflection, to be able to arrive at this concept of intrinsic dignity without theology. So this is basically.
Come la ragione filosofica procede, ovviamente dovendo arrivare ad un postulato, un a priori tautologico, cioè l'essere umano è portatore di dignità in quanto essere umano, cioè la comune appartenenza alla natura umana. Questo è un ragionamento filosofico che non presuppone diciamo la fede in Dio creatore. È ovviamente un postulato monco, nel senso che è ovvio che è un postulato che è difficile da argomentare, però è possibile, perché ogni forma di mancato riconoscimento del valore intrinseco della persona umana, cioè sostanziale, sposta il riconoscimento sugli accidenti e gli attributi, creando forme di disuguaglianza, ovviamente. Che qualsiasi mancato riconoscimento del valore sostanziale dell'essere umano porta a spostare questo valore sugli accidenti o gli attributi, usando la terminologia aristotelica, cioè su valori che sono sottoposti alla contingenza, che quindi possono variare, ok? E quindi creano inevitabilmente disuguaglianza. Questo è un procedimento argomentativo di natura filosofica che non presuppone l'esistenza di Dio, ma che può permettere all'uomo di riconoscere la comune natura umana e il suo valore. Poi l'uomo può essere disonesto e metterci l'idolo, negare Dio con la sua libertà e intraprendere altre strade, ma l'immagine somiglianza di Dio in tutte le creature umane non può essere estirpata. Un ragionamento filosofico onesto può condurre a questo, ed è questo il cammino che bisogna fare con chi non crede e con chi magari non crede in Gesù Cristo o nel Dio creatore, se no siamo.
Okay, I think I understood that. So let me answer the question. Is it possible? Is it possible? Yes, I think it's possible. However, no one, in my knowledge, has been able to do it yet. So if it is possible, I have not heard an argument.
Aristotele.
Oh.
Platone.
Oh yeah. Look, I've read Aristotle and I've read Socrates. I mean, I'm a Thomist, and the question is that here is the question, right? And I'll explain the thing to you. In order to claim that one thing is more valuable than another thing, you need a standard. Even Aristotle had a standard. Aristotle had a standard because he had a first cause. He had a hierarchy of being. So Aristotle, even though he was not Christian, even though he believed in the eternity of the universe, Aristotle already had a hierarchy of being, and he recognized that spirit was more valuable than matter. So even the celestial spheres, the perfection of the celestial spheres. So Aristotle could make an argument for the superiority of the human person because he already had a hierarchy of what was better and what was worse. I am asking, in a secular worldview with no hierarchy at all, how can we decide that the human person is more valuable than other organisms if we do not have a standard to decide which is more valuable or less valuable? If we say that the intellect and the will are more valuable, I ask, why is that more valuable? And if you say, well, it's more valuable because everyone sees it's more valuable. I say that is not true. Because in India, if a cow is more valuable than a human, in their hierarchy, which is a different hierarchy than Aristotle, because their hierarchy is defined by different gods, the cow is more valuable and has more dignity than you or me. So we do not know which hierarchy to use. This is the struggle. This is the challenge. Do you understand?
Okay, this is your break. Nobody wants a break? You want everyone
Okay,
to break?
let's
Everyone who
take
wants
a break.
Those who want a break can go. Those who want to stay.